Welcome to The Pause, a podcast that dives deep into the biotech story and features biotech leaders. I'm one of your co-hosts, Fang Xie.
I'm your other host, Yusuf Baidi.
The podcast is brought to you by the Harvard Biotech Lab, a student-led, Harvard-affiliated biotech organization, which is on a mission to bridge the academia and the industry.
Today's guest is Chris Bahl. Chris is the founder, president, and now CEO of AI Proteins, a biotech company specializing in developing mini-proteins as therapeutics. Chris did his postdoc in David Baker's lab at the University of Washington, where he worked on the de novo design of small disulfide stabilized proteins for the goal of developing targeted protein binders. After that, he started his academic lab at the Institute for Protein Innovation at Harvard before starting AI Proteins and working full-time at the company. In addition to all these roles he has in his company, Chris refers to himself as a protein geek, which will be evident as we hear Chris talk more about his career and work. Welcome to the podcast, Chris.
Yeah, thanks so much for having me.
Cool. Well, first question we want to start off with is we're just interested in kind of hearing a little bit about yourself. Where did you grow up? And now that you're very much interested in protein science, how did you get into studying proteins and into science in general?
Yeah, I think I've just always been a huge geek. So as a Navy family, so grew up kind of everywhere and nowhere. Lived in Boston for a little over 7 years now. That's the longest I've ever lived in one place.
Cool. Well, thank you so much for telling us more. Another thing we want to talk about is kind of what was your motivation for pursuing a PhD and specifically working in biomedical research?
I think I'd like to really maybe credit teachers, right? I think so many folks are doing what they do today because they had a really great teacher at some point in their life. And that's very much true for myself as well. So really great chemistry teachers, really great biology teachers in high school. And that kind of inspired me to go study protein biochemistry. And the more I learned about proteins and how they function, and really that the chemistry of life is mediated by proteins, and they're like little nano robots that kind of do everything. It just became so exciting to study them, to figure out how they work, and then to ultimately try to engineer them to solve problems that humanity faces.
What's your research background? Are you a chemical biologist?
I started off as a biochemist, then became a structural biologist and biophysicist, and then learned protein design.
So what led you to initially pursue a career in academia and start your lab at the Institute of Protein Innovation at Harvard?
Yeah, so my course through traditional academia didn't give me a lot of exposure to non-academic career opportunities, right? I think people in biotech were kind of viewed as other maybe sellouts that aren't doing the good, true work of basic research. And so it's just not something I ever really considered. For me, the people that I held up as being successful that I wanted to emulate were all professors, right? And so that to me was the image of success and career success. And so that's kind of where I pointed myself, not necessarily because I surveyed the career opportunities that were available and chose academia. It was more just the kind of the well-worn path that was in front of me. And I think the world is changing very fast, and people are starting to see those other opportunities earlier in their careers. And actually, I really think it's more moving to Boston. My interests have always been very translational. I think I was always kind of a poor fit in academia. When I was doing my broader academic career search, where to start my lab, the Institute for Protein Innovation was a great fit because it's a translational protein science institute. And I had search chairs at several bioengineering departments tell me that your ideas are very cool and they're extremely translational, maybe too translational for this engineering department. So I think the fit for you is going to be there somewhere, and it's just a matter of getting exposure and finding where that place is where you really belong.
Yeah, cool. Yeah, thank you so much for telling us more about that. And after you had started your lab at the Institute for Protein Innovations, you had the idea to start AI Proteins. So we're interested to hear, how did you get this idea, and what was the process to start a company?
Yeah, I think in many ways it's the magic of Boston, right? I think there are very few places in the world where biotech and academia come together so closely. It's really one contiguous community as well. So, we're located, just up the road from where my old academic lab was. I didn't have to give up my scientific community in order to be able to pursue a job in industry. And that's been really nice. And so the technology that we developed in my lab was funded through research grants and also through sponsored research with venture capital firms and ultimately with biotech companies. And so, we had molecules that we created that were being licensed and going into the clinic. And for me, that was a bit of a realization that, okay, this technology is probably mature enough that it's time for it to leave academia. I had several postdocs that were interested in pursuing careers in biotechs that, were excited about doing a startup gig. And I knew quite a few folks in biotech and also folks that had started companies that spun out of labs. And so I had a lot of a community around me that understood the process and could help provide advice and guidance. So I started fundraising, just going out and talking to VCs, really totally naive to the whole biotech space. And so I think that's also a lot of really kind people took time that they didn't need to spend teaching me about stuff. And so for that, I'm also extremely grateful. This is a community that I think continues to pay it forward in Boston in general and really help each other out. And so this is a really special place.
What was your path from academic scientist to CEO? Many scientists typically aim for CSO roles, as becoming CEO often requires a business or industry background.
Yeah, so I did start as the CSO and became CEO last month. So I did get to learn a lot of how the business works along the way. And maybe actually taking a step back, learning is always fun and learning new stuff. And in academia, it's also kind of interesting where, not to be too cynical, but if you think about how much of your training as an undergraduate really prepared you for life as a PhD student, right? I mean, taking coursework does not really teach you how to be an independent scientist, designing experiments and doing your own bench work and trying to understand, whatever you're studying in your project. And likewise, the training as a postdoc and as a graduate student doesn't really train you for being academic faculty where you're writing grants and mentoring people. So I think what academia actually does a really good job of providing training is teaching you how to figure out how to do a job that you weren't trained to do. So when it came time to go into biotech, I feel like I had done it enough times where I'd gone out and figured out how to do a job that I wasn't formally trained for, that it didn't feel that scary.
That's really cool. Thanks for telling us more about that. And I wanted to circle back a little bit about talking about the ecosystem of Boston and how that was kind of critical for starting a company. So I think after starting your company, around when you started, the Harvard Biotech Club has an incubator program, and your company participated in that. So could you tell us a little bit more about that kind of involvement with our incubator program?
Yeah, for sure. I mean, the Harvard Biotech Incubator was really, for me, a crash course in biotech entrepreneurship. So a friend and colleague and scientific collaborator of mine, Carl Navina, really built that course and teaches it. And so it was really nice that we were brought in and used as the example company for that. And I got to audit the course and follow along and learn with the students as well. And for me, it was also maybe a little bit of learning while doing at the same time. And so it was a great experience.
Yeah, he has textbook.
Yes.
For entrepreneurship.
Yeah, literally wrote the book on the topic. Yeah.
Yeah. Well, thanks for telling us more. And that kind of goes into kind of how you kind of guided or went along that transition from academic to starting your own company. So the next thing we want to know is a lot of people in Boston, specifically grad students and postdocs, are interested in starting their own companies. So what kind of advice would you give people that want to get along that career path?
Sure. So, my particular trajectory is maybe a little bit unique. Having been faculty and then quitting the faculty job, and then everyone from my lab came as well. So we launched with about a dozen people. Actually, one of the department admins came with us as well. She's really stellar. I'm really grateful that she came. And I think maybe the point to make there is that actually, I'm not sure there is a standard way. I think everyone who creates a company is going to have some unique aspect to their journey that brought them to that. And so that's good, right? So, folks maybe shouldn't look for just like a template. Here's where I'm gonna go. 10 people have started companies like this, so I'm gonna go and do that, right? You don't need to follow in someone else's template. So really, the advice is just like go for it, right? If you want to start a company, you need to have a very cool idea. The technology that you're gonna use to build the company is extremely important, and then nobody wins alone, right? It's the community is going to help you. The community of VCs, the community of people that are going to give you feedback on your pitches, the community of people that are going to ultimately join the company and make it be successful. And it's not just scientists. It's the person who's going to do the finances, who's going to be HR, who's going to make your website, right? There's like so many components of this, and it's really a team effort, which is also extremely fun. Being in academia before this, you get really comfortable with this. It's like everyone's an individual, and you can kind of align people to some degree, but at the end of the day, everyone's motivated by getting to be a first author on something, right? And you're not really paid in money because your salaries are so low. You're paid in credit, right? And you need that credit to be able to move on to your next career stage, and that's really so important. And so there's always this calculus in the back of your mind: how much should I help this collaborator? Am I going to be a second author or a first author? And working together, is still not quite the same as being on a real team. And in biotech, that's I think the thing that's been most fun for me is getting to do science on a real team, and people are compensated the old-fashioned way with money, which is very refreshing with competitive salaries. And stock options, right? So when the person at the bench next to you does something amazing, it materially makes your life better. You don't have to worry about competing internally. There's none of that, right? It's like we all win together. The rising tide lifts all ships, and that some ways actually even goes across companies as well, right? Someone who might ostensibly be considered a competitor, their success actually can help you be more successful as well. It's not like academia where the people that publish the first paper scoop you, and your life is sunk after that, right? Like, the most valuable drug on the planet right now is Humira, probably to be unseated by like Ozempic at some point, or Wegovy. But like number 2 is Enbrel, right? And they each make double-digit billions of dollars a year, and actually having multiple drugs available helps create the market. And if a patient starts to have a poor reaction, you can switch them to the other one. So people often say the best thing to ever happen to Coke was Pepsi, right? I mean, that in the business world, having more than one person doing the same thing is actually good. It means you're doing something that's valuable and useful.
Yeah, and I think that also touches upon the point of the point of biotech and pharma. Like, you want to develop transformative medicines to help people. So if multiple companies are developing therapeutics, more people are helped.
Yep, that's right.
Yeah, your TED Talk a few years ago, you mentioned that your company is here, so the AI proteins is focused on developing mini proteins. Could you tell us more about the benefits of mini proteins and compared to other biologics, such as antibody and now it's ADC antibody and other biologics? Yeah.
Yeah, for sure. Mm-hmm. So a mini protein is a long peptide, so like 45 to 60 amino acids in length. And what makes them different than other peptides is that they fold into 3-dimensional shapes. And the mini proteins that we design spontaneously fold themselves up into those 3D shapes. So we create them using generative artificial intelligence. And so we get to control everything about them, their properties, their behavior, their activity. And so we get to really hone them to be ideal therapeutics. And also for applications outside of human health as well. There's applications in agriculture, or diagnostics, many different areas. So when you design a protein from scratch, you have just way more control over what you can achieve as opposed to if you find something in nature. So, maybe if you'll permit a brief digression, I think it helps to an analogy here might help to highlight the technology inflection point. So, in the world of construction of buildings, right? From the dawn of human civilization until pretty recently, we made all of our buildings out of natural materials, things that we found in the environment, rocks and trees, and we would be very clever in how you shape them and fit them together. And we just rebuilt Notre Dame effectively by being very clever about shaping rocks and trees and piecing them together, right? But we didn't make the Eiffel Tower out of rocks and trees, no matter how good you are at fitting them together, no matter how clever you are, right? You need more control and really a technology inflection point, concrete, steel, and glass, enabled the modern construction world. And taking a protein that you find in nature is really what we've been doing since the dawn of biotechnology. Every single product currently that is a protein is something that we found in nature, right? Whether it's the enzymes that are in your laundry detergent, or if it's Humira that's making tens of billions a year every year in sales, it's a natural protein, maybe modified a little bit, like a tree can be turned into a board, but it's still fundamentally a tree, right? So when we design proteins de novo, we're not limited by that. Now we get to make them bigger, smaller, longer, more rigid, less rigid, positively charged, negatively charged, resistant to being oxidized, really susceptible to being oxidized, right? Like whatever you want to do now, you just make it that.
It's really cool. And I was looking more into kind of these mini proteins, and they're a little more stable compared to other proteins. So is that part of it, like maybe targeting disease indications where you might not be able to store them at certain temperatures? Like, is that kind of expanding like what proteins can be used to treat?
Yeah, so mini proteins in particular are very fun. There's a long list of applications where they differentiate. So kind of like what challenge you're trying to solve might influence what the most important features are. But let's take a couple examples, right? So for one is multivalency. So bispecific antibodies. This is becoming an increasingly important area in pharmaceuticals, and so these are antibodies where each arm of the antibody binds to something different. So you can bind to two things and bring them together. Those are often challenging to manufacture and to formulate. The stability can be an issue, and you still fundamentally are limited by having, say, two binding sites in a bispecific antibody, and they're going to be the same distance apart no matter what, because that's what an antibody is. And antibodies also evolved to be part of your immune system, right? And so if you think about when you're infected with something, you want that immunity to be durable. And that thing is going to evolve. The pathogen is evolving inside of you and is evolving then when it goes into your friend, and then maybe you get re-exposed. And you want your immune response to be able to continue to protect you. So polyreactivity actually is selected for in antibodies. It's possible to make antibodies exquisitely selective, but it's actually kind of not that easy every time, because it's a feature, not a bug, for its hundreds of billions of years of evolution. When you're making a mini protein, for example, we design these structures to be extremely rigid. So antibodies, right, are binding with loops. Loops are inherently flexible. That flexibility allows them to adopt shapes that are compatible with many different binding sites. With a mini protein, it's a perfectly rigid little brick. It doesn't flex. You can boil it, and doesn't care. So there's all these like extra, you can imagine now, like, well, if you have an indestructible protein that you can boil and put in acid and sit in digestive enzymes and put in organic solvents, freeze dry, like just mistreat, use, and abuse like you'd never be able to do with a normal folded protein, like all sorts of stuff becomes available. And there are even additional knock-on things that I wish I had anticipated when we launched this company, because we probably could have gotten a better pre-money valuation, but something that we've seen play out because we've drugged like 150 targets in the last 3 years. And we see that they are ridiculously selective for their targets, like way more than I would have guessed. And the best rationalization that I can come up with is actually the structural rigidity confers selectivity, because the protein has no ability to flex into alternate conformations. It has to be in the binding competent conformation in solution, and then it gets to interact with that site. And if the geometry, the shape complementarity isn't perfect, it's not really going to bind. And we see this actually reflected in our binding kinetics. Like we see that, to go sort of deep in protein nerd land, like the K on, like the association rate, how quickly they bind, it's super fast. Like even when we have a really lousy binding affinity, these things come together and bind quick. At antibodies, you actually don't see that. You often find fairly slow on rates. It takes them a while because they're flexible. They got to flex into the shape they need to be in order to bind to your target.
Yeah. But it's like sound like it's better than also better than the small molecule, because you just mentioned like more specific, right? Yeah.
Yeah. So you still want different modalities for different therapeutic indications, right? So with small molecules, the surface area is smaller, and so binding promiscuity is just kind of inherent to a lot of small molecule protein interactions. If you think about it like like a key fitting into a lock, right? If your key has 2 teeth on it, there's just not that many different shapes. There's probably a lot of different locks that you could stuff that thing into. But if your key has like 40 teeth on it, then there's not that many locks that are just serendipitously going to have those exact 40 teeth, right? So another thing, to come back to this point of valency, right? So antibodies, it's pretty tough. You can make bispecifics. There are challenges, but it's very possible. Trispecifics start to become a little bit trickier. You're like Frankensteining parts together, and the developability gets worse. And so with natural proteins, developability, which is really the ability of that molecule to be stable in solution, to be manufacturable, there's all sorts of requirements for protein drugs that natural proteins don't have under evolutionary selective pressure, like sitting in a tube at ludicrously high concentrations for years, maybe, or at least months, right? In order to be pushed through a needle without becoming too viscous. Like all of those things may not be possible when you start to make your biologic more and more complicated. And antibodies struggle with that. So developability and valency are typically inversely correlated when we're talking about immunoglobulin-based proteins. The mini proteins don't suffer from that. So we can just genetically fuse them together like pearls on a string, and then dial the distance between the binding sites very precisely, the order in which they appear in the chain, and like the number of binding sites and the various functions we want those to have. And we've tried over the last couple of years to push the limits of what this technology can do, and like how high valency we can go, and we have yet to find it. So we've made molecules that are 16 valent, 16 different binding activities all strung together, and those are perfectly happy in solution. Each of those binding activities remains preserved. They don't aggregate. And that's just ludicrous. I mean, that's not going into a patient anytime soon. But just like trying to figure out how far can you push it, and it doesn't seem to be a limit.
That sounds really cool. And I feel like going along with the small molecules, like I feel like in small molecule development, there's like the fragment-based drug discovery and combining different kind of parts. So it's really cool how now with these mini proteins, you could have that modularity to it as well to increase kind of the binding efficacy. And then I guess a big part of your company is obviously the use of AI in terms of developing these proteins. And with computational protein design and protein structure prediction winning the Nobel Prize in Chemistry in 2024, there's like a lot of buzz surrounding the use of AI to further enhance our understanding of protein structures. So I was kind of wondering, since you started your company around 2021, how have the advances in AI since then impacted the work AI protein is doing?
Yeah, I mean, it's very exciting to see, and particularly a lot of amazing tools coming out of the open source community as well. So, when we first started the company, we had many of our own, kind of proprietary protein design approaches, just kind of generalizable, like general purpose de novo protein design approaches. And at the time, they were very differentiating. And since then, with all of the people moving into protein design and creating these open source algorithms, whether it's RF diffusion from my postdoc advisor or Chroma from Generate Biomedicine and many others, I think for me a lesson that I learned is that I don't think any company will be able to differentiate based on having a unique generalizable de novo design algorithm, because even if you can create it, the open source community will make something that will be close, and the path to monetization for a medicine is so long that the amount of time that you can hold on to that proprietary de novo design edge is probably at best months. And that's not a meaningful amount of time to recoup a return on investment for all of the, the millions of dollars that you would spend on FTE costs and compute to train that general purpose de novo design algorithm. So I think now it's a bit more of a, what do you do with it, right? What molecules can you create? And there won't be any one company that it just owns protein design and blows everybody else away, right? Much like you're seeing with large language models. OpenAI has a great one, but there are lots and lots and lots of competing large language models, and you're having this whole robust ecosystem, and it's, I hesitate to say gold rush, right? But I think it's less about the specific tool and more about what you build with that tool.
That's cool. And then going along that, like, what do you do with the tool, for instance? I know a big part of the company is like how you, like, with high throughput, are able to produce these proteins and like test them. So could you talk a little bit more about how you guys developed a very high throughput manner at producing these proteins and also testing their functions and activity?
Yeah. So we have built a very large data set. So much like we're not building general purpose protein design algorithms, we have built a very large data set that we're using to fine-tune models to make them better for mini proteins. So we do still need some kind of moat, right? It's very important for any company. And the ability to make them in high throughput, actually, I think is, maybe going back again, is the magic of Boston. So, where I was at the Institute for Protein Innovation really was built to operate around a central core facility, effectively, of shared equipment. And so, when Tim Springer and Andrew Cruz were founding the Institute, it was built around like a $5 million equipment grant from the Massachusetts Life Science Foundation. And so having something like the MLSC, Massachusetts Life Science Center, having the MLSC provide grants to catalyze the ecosystem has been really important. And the founding chief operating officer and one of the other PIs there was James Love. And, James is currently a VP of automation for Novo Nordisk, right? I mean, he's probably one of the best laboratory automation experts on the planet. And I really, myself and people in my lab, we got to like learn at the feet of the master there for a few years on how to build high throughput, like elite protein production methods. Yeah. And it was all really focused around antibodies. And so we got to take and see how you do it at scale with antibodies and then build effectively a parallel process that was focused on mini proteins and taking advantage of many of the key features of mini proteins. Like, we just use E. coli because it's fast and easy, and you want to extract the cells. Like, we use 6 molar urea as a lysis buffer, and we sonicate them without cooling, right? So, for the non-protein nerds, listening, it's just like a super harsh denaturing buffer that would obliterate natural proteins, and then you cook them under harsh conditions at fairly high temperature. Like, these conditions literally dissolve the entire cell. And our mini proteins survive that. And so you can leverage that sort of thing to build what we've done, kind of pharmaceutical scale, like big pharma scale, high throughput protein production. Like, we were actually really close to this throughput you find at like Pfizer's high throughput protein production facility, but we're a little startup. And that's possible because of the I would say what we learned about how to automate various steps and also exploiting kind of the ridiculous properties of the molecules that are unique to us.
Cool. Well, thank you so much for telling us more about like kind of the platform and everything. And I guess as the company evolved, I know you guys kind of considered other applications of like these mini proteins. So I don't know if you could tell us a little bit about like maybe partnerships or kind of use cases that have come up, in the past 3-4 years.
For sure. So, we announced last month a biobucks deal with Bristol Myers Squibb. And, for any company with novel technology, having a big pharma come in and publicly say, we want what you're building has been, huge for us, right? Very important. And also just really excited to work with a company that has so much credibility in being able to bring drugs to market, right? I mean, we know that those molecules are going to be in good hands. And really, I think the platform we've created here is a bit unique. I think over the last couple of years, we've seen in the biotech venture capital community a deprioritization of investing in platform companies. And my friend Yushini Scarlett just published a paper, an article in BioCentury during JPM, where she actually quantifies and shows that investor sentiment, investor, I guess, interest, and where they're deploying dollars, whether it's a platform company or an asset company, is harmonic. It's quantifiably harmonic, and so you can see that everyone's scrambling towards platforms. Everyone's putting money in assets, and then it goes back and forth, and it just oscillates. And so, we're swinging back into investor interest in platform companies, and I think for us now this is a great opportunity because we're really a platform, right? We can bring drugs into the clinic, and we do plan to, but we've drugged about 150 targets in 3 years. I mean, that's bonkers. And we're still just getting started. So that's way more than we could ever bring into the clinic. And that's more than like Pfizer can bring into the clinic, right? So even with a robust ability to partner, we still have more discovery capability. And so this, I think, unlocks a business model that is not common for biotech companies, which is to truly be a platform. We look at maybe another company out of New England, Adimab, right? That's in Lebanon, New Hampshire, and they're really the platform that is the most platformy platform to ever have platformed, right? I mean, they have like a huge number of drugs that they've made that are in the clinic. And I think something like that is very exciting because they get to stay true to who they are. You hear these, it's a tale as old as time. You have this great, unique platform. It makes a molecule that gets to late phase 2 or phase 3 clinical trials, and then the investors push all the capital towards finishing the clinical trials, and the platform gets thrown in the dustbin, right? And so really the goal here is to have a big impact on humanity, to do a lot of good. And we really want to be able to keep the platform intact so that it can keep creating molecules, keep creating value for our investors and for our species, because we can solve problems in agriculture and human health and you name it. We don't want all of that to get thrown in the dustbin when one asset gets close to an FDA approval. So for us, a big part of our business strategy is also to create new companies that will effectively be partners that will take our assets forward. There are a lot of people in the world that are very smart and very good at running clinical trials and can do drug development. We're really the only ones in the world right now in the biotech space that can make mini proteins. And so we want to be able to keep continuing to do the thing that we do best in the world and then partner with people that can do the other thing that we can't do.
Yeah, that's a good point. Kind of the harmony that needs to exist between platforms and kind of asset indication companies. Because, for instance, for CRISPR, it's like there are a lot of companies just focusing on that, and now we've seen like large pharma companies use those and develop them for clinical trials. So I think the same thing needs to be done for other modalities, such as like the mini proteins.
Yeah. For mini protein, I'm very curious about that. For mini protein, do you think that's also can go through the brain blood barrier? I mean, would be possible? I mean, for mini protein.
So maybe. So there are mini proteins in nature. They typically are disulfide rich, and actually the very first mini proteins that were designed de novo, I did as a postdoc when I was with David, and I made those disulfide rich as well. And that was simply because I got to David's lab kind of at the dawn of de novo protein design's reemergence. It was done the first time, top 7 in the early aughts. Brian Coleman, the first author there, his landmark paper, and then it basically wasn't done again for a decade. And so when I joined the lab, they were just kind of getting it going again. And David was basically just like, hey, what happens? How small of a protein can we make? And that was my introduction to mini proteins. So just try to look and see what the smallest folded protein in nature was. You find them in like venoms, often like scorpions and spiders, and they use disulfides to like staple the structure and make them stable. So that's how I made the first ones. So this is a long-winded preamble to say there is a natural mini protein called chlorotoxin. It's isolated from a scorpion venom, and it serendipitously binds to cancer cells, many different types of cancer. We don't really know what the target is. There's a couple papers that claim to have identified it, but those data have been tough to reproduce. It's probably a protein-lipid interface. And importantly, the molecule also binds to the liver. So it's not really possible to warhead it with a cytotoxic payload. But Jim Olson now at Seattle Children's Hospital attached a fluorophore to it, and then you can inject this into a patient, and this is particularly important for patients with glioma and glioblastoma, right? Because you are oftentimes surgically resecting these tumors, and in particular, he's a pediatric oncologist, right? So, so important when you're doing the surgery on this. The margins need to be extremely precise. You don't want to take healthy tissue. You can really impact someone's life, and you don't want to leave anything behind because you can't go back. And the time spent sending samples to pathology are critical moments when you have that surgical field open. So being able to see the tumors in situ is really awesome. So Blaze Bioscience got accelerated phase 3 status for their molecule they call tumor paint. And that does seem to cross the blood-brain barrier, and so you can see these. The tricky thing is you can't really tell if it crosses the blood-brain barrier in the absence of cancer because it binds to the cancer, right? And brain cancer also is well known to make the blood-brain barrier a little bit leaky. So
Yeah, that's like an interesting caveat. It's like how functional is the blood-brain barrier in glioblastoma, and maybe that's the special case where it actually does cross it.
Yeah.
Cool. Well, thanks so much for talking about science. And in terms of like community, specifically like in the Boston area, we see you're the founder and co-organizer of the Boston Protein Design and Modeling Club. Could you share with us how this group started and what it's been like to continue being part of this community as you made like the transition from academia to industry?
Yeah, for sure. Yeah, so I love BPDMC very much. So it actually started off as Rosetta Club.
Oh yeah.
So when I moved to Boston in 2017, back in the bad old days of protein design before AI really was making its mark, Rosetta was kind of the bee's knees at the time, and that software I think is. While I love it, and that was my formal postdoc training, I think the best way to describe it is user hostile.
Yeah,
it's exceptionally hard, and so it's very, very rare for anyone to really learn how to use Rosetta unless you trained in a Rosetta lab. And so it's basically diaspora of David Baker that know how to use it, or second gen diaspora kind of thing, right? So when I moved to Boston, there were no Rosetta PIs in town, and so there was no one who really knew the software that anyone in the city could talk to about protein design. And so when I moved here, I found just like a really excited community of people that wanted to design proteins that had been struggling with Rosetta. And so it actually started off with just going to the bar, actually Squealing Pig next to Longwood, and just like getting oysters and having beers and just like talking about troubleshooting scripts. And it was clear there was a tremendous amount of enthusiasm for that, and people that wanted to learn more. So then we started, turning it into. Originally, the first formal meeting was I did a tutorial on Rosetta scripts and like how to select residues and a little more advanced, Rosetta features. And then a lot of the people that were working with Rosetta, we wanted to be able to help them more, so we almost turned in like a lab meeting. So people would come in and present research in progress and could get feedback from myself and others. Sergei Ovchinnikov also moved to Boston, first as a John Harvard Fellow, and now he's a faculty at MIT. And he was really more on the hardcore protein structure prediction side. Folks, other modeling and design folks starting to get more involved. Actually, Debbie Marks was the inspiration for changing the name from Rosetta Club to the Boston Protein Design and Modeling Club, because she very astutely pointed out that not everybody uses Rosetta. And very grateful for that as well, because we wanted to make the community very inviting and inclusive. And now that Rosetta is kind of also on its way out, it's nice that we have a name that is not tied to Rosetta. And so Sergey first, started helping me co-organize, and then now Nick Polizzi, who's at Dana-Farber and trained with Bill DeGrado, kind of also not necessarily directly from the Rosetta community, but still very close to it. Came in and when he moved to Boston too, I kind of recruited him in to help co-organize, and so that it's really great to have the anchor on both sides of the Charles at MIT and at Harvard Med. In academia, and then I try to help out as well. It's expanded tremendously. I think there's about 700 people on the email listserv. So during the pandemic, we went online, and after the pandemic, we now are fully hybrid. So we we broadcast to a global online community, and then we also have a pretty good sized group of folks that show up in person. And so, it's both science and then also networking. And now there's VCs that are paying attention, lots of big pharma, lots of biotech. So I'd say it's probably split roughly 50-50 academia and biotech. So it's a great ways for all scientists to mingle. And it just, again, I'll just keep saying it, it's the magic of Boston. I don't think that kind of thing really happens to the same degree in other places because nowhere else has the kind of density of nerds that Boston has.
That's true. Yeah, a lot of protein geeks in Boston, and it's really cool that you made it accessible to people outside of Boston as well to attend virtually.
Yeah. Chris, I think that our listener audience may be very interested in hearing about if you have any suggestion for the students or residents, fellow postdoc who are currently now in academia, but maybe want to want to do a startup company or join a startup company. Do you have any suggestion?
Yeah, I mean, getting involved, right? Showing up. There's lots of places to learn about it. Harvard Biotech Club is a really fantastic place to start getting exposed to it, building your network. And there's also Nucleate is another good place. Having really cool technology is fundamentally going to be important for any biotech. So it's not all about hobnobbing and meeting people. It's also about doing something that's really transformative, has transformative potential. And it doesn't have to be a fully realized idea, right? That's, I think, one of the other very cool things about biotech and the startup ecosystem is you can just have a great idea. And really at any stage, you can find someone willing to support your idea. You can do a small pre-seed round if it's just total blue sky. And if it's a compelling enough an idea, you can just get started on it. You don't have to wait.
Cool. Okay. Yeah.
Cool. Thanks. Well, I know you have a meeting to run to after at 5, but we want to thank you so much for taking the time to talk more about your career journey and more about AI proteins. And I think people are going to be really interested to talk and reach out to hear more about kind of the company. So it's great to hear that you're kind of accessible and able to give advice.
For sure. Yeah. Love chatting with folks. Yeah. Thanks so much for the invitation to join you guys today.
Yeah. And thank you so much for also the continued support of the Harvard Biotech Club as well.
Yeah.
Mm-hmm. For sure. I mean, you guys were essential in my own entrepreneurship journey and helped me launch. So, I would love to help pay it forward back to this community that's helped me so much.
Cool. Well, thank you, Chris.
Thank you so much. Yeah.
This is episode 2. A big thank you to my co-host Yusuf for planning the show and co-hosting this interview with me. And thank you everyone for listening in. Our next guest is George Church, professor at Harvard Medical School and the Wyss Institute. Stay tuned. We will see you in the next episode.